quite a common design that I see in particularly agricultural research is, let's say, a case where we've got an on-farm trial with a bunch of different treatment groups, and we're looking to investigate the effect of crop cover and nitrogen application on the yield of some kind of grain. This particular example, we've got 9 farmers, all of whom are applying all the 8 different treatments. So once we get our data together from this particular design, I'll often see plots that look something like this, where we have our 8 treatment groups and we have one bar representing the average value of the yield for each of these 8 treatment groups. But there's actually quite a lot of limitations to this particular way of displaying the information. Fine, we have all of the average values correctly placed there, but we're missing quite a few ways to improve the utility of this plot to really drag out the key results and the information we have from our data. So if we just go back to our 8 treatment groups, the key thing to identify is we don't really have 8 treatment groups. We've got 2 different treatment factors. We've got one with 4 levels and one with 2 levels. So we've got 4 different crop cover treatments, 3 different kinds of crop, and then one with no crop cover at all, with continuous maize. And then we've got 2 different kinds of nitrogen, whether it's applied or not applied. So with this sort of structure, we're really losing information by not capturing that in the way that we plotted our previous graph. We want to respect this structure by trying to understand, okay, is there an effect of crop cover? Is there an effect of nitrogen? And is there an interaction between the two? Which is not just looking at the 8 individual values, we need to add some structure to our plot. So even just taking the same 8 mean values, we can put it together in a slightly different way. So grouping together the nitrogen applied and grouping together the nitrogen not applied, using different colors for our different 4 treatments. And previously in the last bar chart, we were really struggling to pick out any big trends. We could maybe find the minimum and the maximum average yields, but we weren't clearly seeing any patterns. Whereas now we can see, okay, generally with nitrogen being applied, the yields are higher on average. But there's one of the treatments, this blue bar there, the crotalaria, where actually getting higher average yields when nitrogen's not being applied. And we can also see, for example, our Artifonia and Tifrosia, the purple and the green, they're very, very similar yields when nitrogen's not applied. And then again, when nitrogen's being applied, they've got very, very similar yields. So these are 2 very, very similar looking crop cover treatments. And with the continuous maize treatment, this is the one which seems to be getting the biggest benefit of having the nitrogen being applied, because we see it's similar to the purple and the green when nitrogen's not applied, and the average is quite a bit higher when it has been applied. So by restructuring just those 8 bars, we've immediately made a more informative plot by factoring in our 4x2 factorial structure. In fact, we could make that even easier. I still had to sort of think a little bit when I was going through and looking at those bars. If instead we use points and lines, so this would be commonly known as an interaction plot, all of those points that I just mentioned when I was looking at the bars, they become much clearer when we're seeing, okay, yes, the increase from not having nitrogen to having nitrogen, the similarity between the purple and the green, and this strange thing going on with the crotalaria decreasing between having nitrogen not being applied and nitrogen being applied. When I was deciding how to decide what goes where with this plot, I also respected the fact that with the nitrogen treatment, there's an inherent order to this. We're going from nothing to something on the x-axis. Whereas my 4 crop cover treatments, these are just 4 different things. There's no order in my 4 crop cover treatments. So it made sense to put the nitrogen factor on the x-axis and use 4 different lines to represent my 4 different treatments. And this principle can always be applied with any kind of 2-way interaction. When we're having multiple factors and we want to understand overall, is there an effect of one of the factors? Overall, is there an effect of another factor? This is looking to see whether we're having values increasing along the x-axis, and then looking to see if we're having separation between the values in the different lines that we're drawing, but also the interaction. Are these 4 lines parallel to each other? Looking at the data that we've got, with the plots I've made, my initial reaction would be, okay, it's very likely we have a significant effect of nitrogen, a significant effect of the crop cover, and a significant interaction based on everything that we're seeing so far. But there's a bit of a trick here. In fact, none of these results were statistically significant if I went ahead and did my analysis of variance. And this is kind of a key point that we should always be considering, as well as the structure that we have within our data. We also want to be respecting the results that we're finding from our analysis. So inevitably, one of them was always going to be the highest and one of them was always going to be the lowest due to variability. But we always want to have some degree of uncertainty or confidence in the results we are presenting, because they are never just a singular number. They represent an estimate. And the trends were quite overwhelming in those plots. So it's a little surprising to me, perhaps, that these are not statistically significant. And this is because of how much underlying variability there is in that data. So we could see when we were just looking at the bars, all those average values were around 1 to 1.5 up to 2 tons per hectare. But within the 9 farmers that we had, if we look at a box plot instead of a bar chart or any kind of plot that only looks at the mean values, and we can see the range of values goes all the way from pretty much 0 up to 5 tons per hectare. So this level of variability in the data means we can't be confident in those differences that we're observing. So we need to make sure we're not misleading people with how we choose to present our results. And in a separate video to this, we'll talk more about the uncertainty and the way that we can represent uncertainty around estimates with standard errors or confidence intervals and things like this. So in general, when we have this factorial data, there's a few key principles to bear in mind. Firstly, we definitely always want to be matching the structure of the data to the structure of the graph. In this case, we only had 2 factors, and we could have one as different lines and one as our x-axis factor. It can also really easily extend this with having little panels. Sometimes you hear these called small multiples or facets. These can bring in a third and even a fourth factor by just designing a graph which has a structure which matches our treatments. If we start getting more than 4 factors, this is a bit too much information for the human eye to be taking in just looking at one plot. So we might take averages where it makes sense to take averages and having multiple plots to highlight different trends. And we always want to be trying to use the benefits we have in our data. If there is an order in our data, then use that as a factor for the x-axis. Similarly, if there's some kind of convention around using colours for particular groups, if these are different plants which are different colours, try to make sure that you respect the visual metaphor by making it as easy as possible for somebody reading this to understand how all these components fit together. Generally, bars are not a good tool when we're comparing multiple factors. And it can also be quite easy to make misleading bars where you can make quite big differences look quite small, or quite small differences look quite big, just by how you're drawing the bars. And using lines, using these interaction plots, draws the eye to the comparisons across groups better than a bar. The bars may be easier to read off against the y-axis the exact value, but we're nearly always more interested when we're looking at a factorial experiment of making comparisons across groups. And using lines is a much better way of drawing the eye to this. And don't be afraid of the variability in the data and the uncertainty around the estimates. You don't want to be misleading people by presenting results and saying, yes, this is my best treatment, when you know separately to this, you've done the analysis to show that that is not a conclusion that you're able to support. So make sure the plots complement the analysis rather than contradict the analysis or confuse the analysis. And the final point is, don't be afraid of trying lots of different things. The plots that I've shown here, these are not the first thing that I create. It's a bit of trial and error of multiple options to see what is working best for highlighting those key trends and patterns to make it as easy as possible to communicate your results. And don't assume just because you've tried something before, it's necessarily going to be the best thing again. It's a good starting point probably, but always seek to do a bit of trial and error to try and make your plots as nice as you can.